If you turn to the book of Romans chapter 1, we'll start there in just a second. Romans chapter 1. I do appreciate being with you this evening. It's been a great series. I went online and just saw what have other people done. What have you already heard to this point? And it's a lot. It's a lot. And it's needed. We live in a culture with a message that is exactly the opposite, of course, of Scripture, that God is, that His message is, and that we can understand it. It's knowable. And we can all understand the same thing. And we're told the exact opposite of that all the time. And I hope to do a little bit again this evening to help answer this question for all of us. Why believe? Why believe at all? So in Romans 1, I'm sure you've heard this verse more than once this summer. We'll read it again. Romans 1, beginning in verse 20. For since the creation of the world, His invisible attributes are clearly seen, being understood by the things that are made, even His eternal power and Godhead, so that they are without excuse. Because although they knew God, they did not glorify Him as God, nor were thankful, but became futile in their thoughts, and their foolish hearts were darkened. Professing to be wise, they became fools, and on the rest of that chapter goes to explain the fall of culture from intellect. Decided, I know more, and so I don't need God, and we can move on with that. There's loud voices in our culture that speak opposite of what this verse just says. That you can look in nature, and God is talking to us in an unmistakable voice, an unmistakable speech. And Psalm 19 pictures it that same way. Day unto day utters speech. Night unto night reveals knowledge. God is speaking to us. We can't know His will from nature, but we can know that He is there from nature. And then He has spoken to us in His Word, and that's where we learn His mind and His Spirit, what He really wants us to do and wants us to think. So we're going to look this evening at a couple of things. We're going to look at a case, one in particular, of God's power. Is that on? There it is. Of God's power in creation, in particular, God in sight. Probably not. Let me see if I get it. There it is. There it is.
You're a chemistry professor.
This just didn't show up. I can see it there.
No, that's
Got
right.
it.
It'll be back there. Sorry.
No, you're good, Josh. It's like there's nothing on the screen. And oh, there it is. There it is. The screen of antiquity. It once had a purpose. It's vestigial, I guess. It's no longer needed. We're going to take a look at vision. It's an old argument that look at the eye of a human, and it looks remarkably designed. We're not going to actually look at the eye. We're going to go farther than that. But God is in sight in more than one way. You look at how sight works, and God's right there to be seen by it. He's not hidden. God isn't something we have to discover. He's right there, near each of us, that we will grope for Him and find Him. That's how He's always been. Back when Darwin was growing up and being educated, there was a book that I think Buddy mentioned last week called Natural Theology, and it was required reading in Darwin's education. And one of the arguments in that book was the famous watchmaker argument. Look at a watch. If you were to define that, you would conclude something designed it because it's really complicated. And that was a really common argument, and a good one, by the way. And Darwin grew up with that. Now, there's a whole other study about one reason why he sort of pushed that argument aside, because the way it was framed was actually fundamentally flawed in some ways, and it's a whole other lesson, a really good one. But it sort of set Darwin up to say, well, that doesn't work. Let's go this other direction here. So in 1831, I think, Darwin set sail on the Beagle, spent 5 years going around the world. You know that story. And he got back and started mulling over his theory. And in 1858, he published this paper, which was his first public declaration about his theory of natural selection. That's how this variety of species came to be. A year later, he published his book, Origin of Species, sold out in a day or two in Europe, and the world has never been the same. Since Western world has never been the same. It is a profound impact that book has had on every aspect of culture. It dominates not in just natural science, but all the social sciences. It's everywhere. And in that book, this was Darwin's key idea. The key idea was populations change over time, which everybody knew. Nobody argued that. But populations change over time due to natural selection. And the mechanism was the key thing. Not everybody accepted that populations change, but most everybody could, and you could see it happen. They'd seen it for thousands of years. It was the mechanism that was unique and gave an intellectual validation that you don't need God anymore because natural selection can do it. And that's the debate we've had ever since. And not everybody, including his pals, bought into that entirely at the beginning. One key spot in that formation of his theory, is the Galapagos. If anybody knows anything from origin, you probably know he stopped in these little small archipelago of islands off the coast of Ecuador, and he saw all kinds of things. Maybe most famously, he saw the finches there, the birds. And he looked at their beaks and noticed, did God really create all these types of birds, one for this island, one for this island, one to feed on this plant on the island, the other to feed on this plant on this island? Is that really what God did, or did birds get there, and over time they diverged, which is almost certainly what happened? Well, if birds get different sized beaks, then maybe bird came from a non-bird, and that came from something else, right? And he came up with this famous tree of life. The only picture in that entire book, if you ever want to go to sleep, read Origin of Species. It's a hard read. It's hard. One picture, and it's this one. And that's his version of trying to illustrate his thought. Right? All living things that we see today have branched off some sort of common ancestor, and that's the dominant story. And it's a story of simple to complex. So when somebody says, look, something like the human eye is really complex, then the story is, well, that's right, but it didn't always start that way. Make that eye just slightly less good, but it still works. And make that eye slightly less good, but it still works. And keep going all the way back, and eventually you get the first version of an eye, which is a one-celled organism that could detect light. That was the start of it all. That's the story. And it can sound very convincing, and it can look convincing with things like, well, you start with the eye. I'm going to skip this quote. It's things like this. It's a very simple eye on a limpet, a very simple animal. Has a little layer of cells that includes some cells that are light sensitive. And that's connected to nerve fibers. I say that's simple. That's actually profoundly complicated, as we're going to see in a second. But as far as eyes go, it's pretty simple. And then you have the abalone, which has the same kind of cells, but now they're arranged in a cup. So you start to have more structure to it. You can start having a little better idea of where is this light coming from. You start getting directionality because the cells are oriented differently. One's pointed this way, the other's pointed this way. You get a little more complicated with the nautilus. Now you actually have a fluid-filled cavity that covers those cells. And then you get snails, some snails anyway, and they actually have a lens over that fluid. And then you get a squid, which has an eye very much like yours, very complex, very high level of vision, high definition of images. And you lay this story out in pictures, and it's a story. Well, it's supposed to be a story. It's pictures, and it's only a story. That if you can go from this simple eye, and then a little more complicated, a little more complicated, a little more complicated, that's where I came from. Folks, that's a powerful story with those kind of pictures. That has a powerful communication to it. But it's just pictures. Pictures without a mechanism are no story. They're no explanation. They're pictures. And you got to have a mechanism to it. How did that happen? And to say natural selection did it is no mechanism. It's a story. You got to get down to where life happens. And life happens on the molecular level. I think Buddy talked a good bit about that last week with DNA, right? We're not going to get quite that technical tonight. I saw his slides and I thought, they probably have had enough. There's about 5 of you who are in my classes who might say, all right, let's give it. The rest of you are like, bleh. But we're going to get a little technical. But then step back and ask, why do people have a hard time even allowing God in the door? Why even allow God in the door? Rudyard Kipling wrote a set of stories. Some of you may have read them to your kids. They're called Just So stories. They're really fantastic stories. They're filled with made-up words, and it's things like how the giraffe got its neck, how the elephant got its trunk, how the camel got its hump, and all these and they're just fantastic tales for children, but they're just so stories. It's a story that could make sense, but it doesn't. At the end, that's just how it happened. That's what this is. This is a just so story. You got to get down to what actually makes it work. That's what we're going to try to do this evening. What does it take to actually have not just the complexity of a human eye? Let's go back to the very beginning. What does it take for an organism that's one cell big to detect light? What does that take? So we're just going to talk about the simplest kind of mechanism that it takes to detect light at all. First of all, to have an advantage, an organism can't just detect light. It needs to, but it needs to do more. It has to be able to sense light versus dark. That's true. But it has to communicate that information to itself. Who cares if a light can react, if one of your cells can react to light? If none of your cells are affected by that, so what? Do you move towards it? Do you move away from it? Can you make energy from it? You got to be able to react to it. You also have to integrate that information, hey, there's light coming from over here, with all the other information coming from your environment. You and I don't react to one kind of stimulus. Right? It's what we see and hear and smell and feel, and it's the combination of things we have to integrate to make a decision, and so do even little single-cell organisms. And then fundamentally, we had to do something. There's light over there, but unless I can react, who cares that I can know light is there? I have to be able to react for it to be beneficial for me to even sense it. And then whatever system is that lets me do all those things, I have to maintain that. It has to work right and keep working right. So how does that happen in a single cell? Well, if you were to take everything that's alive and try to organize it, you would end up with three big buckets, three big files. The green one is eukaryotes. That's you. Those are the most complicated kind of cells. We call them eukaryotes. Don't get too excited. We're in there with algae and fungus and all that kind of stuff. Complicated cells. The next group we're familiar with is bacteria. They're only one cell big, extremely complicated. We've never built anything remotely as complex as one of those cells, but they're not as complicated as you by a long shot. And then there's this red group that people for a long time thought were bacteria because they look just the same under the microscope, but when you get down to chemistry, they're actually different. And they called them archaea. Sounds like archaic, right, or old is the idea. And they have shared a lot of similarities with bacteria, but they actually share some similarities with eukaryotes as well. We're going to take a look at one of those critters. It's called a halobacteria. This little oval-shaped guy, best picture I could find, is a little fuzzy. They're really small. They have a little flagellum on the back, which rotates like a boat rotor. It's not a tail. It's not a tail. Tails don't rotate 360 degrees, at least not often. They break. They don't wag that thing. It rotates like a boat rotor at thousands and thousands of RPM, tens of thousands of RPM. In fact, they can rotate that thing at tens of thousands of RPM, stop it, and rotate the opposite direction in less than one turn. It's remarkable engineering, the way that thing works. That's what we're going to look at. This little bacteria likes to be in certain kinds of light, doesn't like to be in other kinds of light, so it needs to know where light is. And the way this thing moves is fairly random. You see on the left side, if there's no light stimulus from the environment, it just sort of randomly meanders around, like a lost freshman. Where am I? I don't know where I'm going. I'll just walk this way a while, like, I don't know, maybe I'll try this. Right? And that's what it does. So it'll swim, and then the flagellum will stop, and it will just rotate in three-dimensional space, and then it'll start rotating again. Whatever direction it's pointing in, that's where it'll go. If light is around, however, it changes either how long it's swimming in the right direction, if the light is good for it, because there's just like you and me, some light burns you, right? Same thing with these little guys. If it's good light, it'll swim longer before it tumbles around. If it's harmful light, it'll tumble faster. It'll tumble soon because it'll realize I'm not in a good spot. I need to be drifting somewhere else. And so you have what's called a run and tumble movement in these little critters that do their thing. And it's capable of responding to those kinds of stimulus. So how does it respond? Well, here's where all the cool people get excited. All right, this is a cell membrane. Right? All the cool people like that. The outside of every cell looks very much like this. You have two layers, these little things, like with a little ball with two legs sticking on it. And there are two layers of that. And everything that keeps the cell together. That's like the skin on the basketball, is that thing. It's called the cell membrane. And stuck all over it are proteins. Those little pink bubbles in the picture, those are proteins. So those are all different kinds of proteins, and they have all different kinds of jobs. So for now, just know that. The outside of cells has protein stuck on over. I'm going to teach you 3 proteins. It's only 3. Only 3. Business majors, you can relax. It's only 3. It's only 3. But they're critical to get this process going, to make it work right. The first is called retinol. This is a stick figure of chemistry. That's what this is. When you take organic chemistry, you don't want to draw those things out, so you just draw stick figures. Everywhere 2 lines join together, you have a carbon atom, is what that means. And other stuff's built on it. You're built of carbon, is what it is. And there's a key point on this little molecule right there. There's a little joint that normally, if I were to depict it, it's like this. Your hand is up. But when the right kind of light hits it, it just pivots down. And it moves from that position to that position. May not seem like a big deal, but that sets off a domino effect. So when a particle of light slams into this molecule in the cell, it just doinks, for lack of a better term. Right? It goes from here and doinks to there. All right? It moves. First protein, we're done. Retinol. Good? Not hard. Second protein is called sensory rhodopsin 2. Sensory rhodopsin, no problem. If you look at this picture, you see a little pink squiggle right in the middle. Right? That's the retinol I just talked to you about. So retinol is kind of a small little molecule, and it's buried inside this big protein, sensory rhodopsin 2. And that protein is wedged in the cell membrane. See the little head with the 2 little legs sticking off of it? Right, that's the outside of the cell. And this protein is wedged in there, and it zigzags up and down the cell like thread, like you threaded it 7 times. And those little spirals sort of tell you how it's arranged. And those are actually chemically really important. And the way they arrange themselves is really important, beyond the scope of this talk, but they do that for a reason. To my students, I say, it looks like a telephone cable. Then I always these days stop and say, how many of you have ever seen a telephone cable? And I actually have one that I bring to class now. I say, this is what a telephone cable used to be. I can bring it with your TV. Archaic. It's one of those things. And so what happens when retinol does its doink, when light hits it, it's connected to sensory rhodopsin 2 in a very specific place, in a very specific way. And when it moves, it causes sensory rhodopsin to change its shape through a series of steps. It's sort of like a click, 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 click. As it clicks around in microseconds, it clicks like that. And they have names for that. You don't have to worry about that. But as you see a little blue bolt of light coming in at the top, it hits sensory rhodopsin 2, and then it shifts to something called the K position. It takes then to the L position, then to an M1, an M2, an O, I think, as it says, an O position, and then it finally resets itself. Right? So it runs through this very, very fast chick, 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 chick thing. And it's a protein that's moving on the surface of the cell. And the key moment is that M1 box. The M1 box is the signal moment, where it sends a signal to something else. So you've had light come into the cell, it hits retinol, retinol swings its elbow down, it makes sensory rhodopsin 2 do its cycle of changing shape. Right next to rhodopsin is protein number 3. Protein number 3. And that's called HTR protein. And it sits right next to it in the cell membrane. So when the rhodopsin starts changing its shape, it senses that. It can see that. So you're communicating right here on the cell. Light has turned into a form of communication. And the cool thing about this protein is it has part of itself sticking out, the red, sticking outside the cell. And all that green part sticks down inside the cell. That's like passing a baton. That big green tail is way down inside the cell, so if something happens up outside or in the membrane, the cell can figure it out down inside. Not just anything can come in and out of the cell. You want to be protective of that. So how do you communicate something that's happening outside, like, hey, light just hit me? How do you get that down inside the cell? Well, you need this protein. So that when retinol swings its elbow, rhodopsin does its cycle of clicking, HTR picks that up, and the tail down inside starts wagging around and carries the message deep down inside the cell. It's remarkable how that works. Just keep it easy. One more picture, because it's just cool. The red is the rhodopsin. The green is the HTR protein, and they line up in pairs. two rhodopsins, and right next to them two HTR proteins, and they communicate with each other. And if you take this picture and pivot it so you can look down the telephone cord spiral, it looks kind of like this. And you'll notice that I don't know how well you can read the letters on that screen. They're pretty small. But one of those spiral red pieces is really close to one of those green spiral pieces. And they talk to each other. If you zoom in, it's right there. That's what I'm talking about. See that on both of them? The green and red piece are really close together. That's where they communicate. And they have to be lined up the right way for that communication to happen right. And if you zoom in really close on one of those, there's all kinds of other stuff sticking out. This is chemistry, right? It does. And you're right. The chemistry are kind of they're kind of the weird ones. Yeah, they're kind of weird ones. My first job was teaching high school chemistry, so I can say that. And they communicate. And so you have this remarkable system where a cell becomes sensitive to light. Well, does a cell just become sensitive to light? You have to have the retinol protein produced, which means you have to have that gene in your genetics. You have to have sensory rhodopsin produced. That gene has to be in your DNA. It has to get put in the cell membrane in the right way, in the right orientation. Retinol has to get stuck inside it, attached to just the right part of that protein. It's made of hundreds of amino acids. Proteins are all made of amino acids. Retinol is attached to a specific one. I think it's number 216. It has to be attached to that one. And then next to that, you've got to have HTR protein, which has to be in your DNA. It has to get to the cell membrane and orient itself just the right way so it can listen to the rhodopsin talking to it. It's remarkable that can happen at all. And when it does, you get a cascade that I'll just go through fairly quickly of the kind of proteins. So you have light hits the SR2. Buried in that is the retinol. Right next to that is the HTR protein. And when the HTR protein starts wiggling that big green tail down inside, it talks to something called CHEA. And it has to have help doing that from something called CHEW. And what it does is it makes CHEA grab something called phosphate. When a cell sends communication to itself, phosphate is a really common switch. It'd be like if I took a little Post-it note and I just stuck it on you and said, get this message to the back. Well, you could take that Post-it note and stick it on you, and you could take it and stick it on you, and you could take it and stick it on Buddy, and it could make its way to the back. That's what phosphate is to a cell. You can pass phosphate around. That's what happens. CHE says, well, I need a phosphate. So it grabs the phosphate. Now it's CHEA-P. And it takes that, and it sticks it on CHEY. So now that has the sticky note on it. CHEY now can go and talk to the switch on the motor that drives that flagella, that thing that spins around, because that's all controlled. It's highly controlled. And that then affects how much that little bacteria cell either keeps swimming forward or stops the motor so it can tumble and get out of Dodge and go somewhere else. Incredible. Take any of those proteins out, that doesn't work. Who cares that you can detect light? Who cares that HTR is wiggling its little tail down in there? It doesn't help the cell to do anything. It's not going to. It can't react. All that has to work right, and all those proteins have to function right, and they all have to be in the DNA all at the same time. And of course, you have to reset this whole thing. So you see in part of it already, the CHEA with the phosphate. Once it dumps its phosphate off, there you got the CHEA back again. But there's other parts that do this. You got to get the P off the Y. Well, there's another CHE protein that does that. There's another little one, CHEB, that helps pull the phosphate off of the A. There's another one called CHER that talks to the HTR protein. In fact, so does CHEB. Don't you see how much fun cell biology is? Right? It doesn't take long before you're like, oh, whatever. I don't know what's going on. Hannah Cooper's in for a good ride. She's in my class. I'll see you Friday, Hannah. It's going to be good. But all this is part of the control. And there's more layers of control to that little purple HTR protein. You can stick things on it that make it more active and make it more sensitive. You can stick other things on it that make it harder to activate. So you you have a brake and an acceleration pedal for this whole process that's separate from light completely. It's amazing what it can do and how much that can work. And so you do all of that just to be able to react to light at all. And in addition to that, you have to reset the whole thing. You have to integrate all the signals that cell is getting, not just light, but chemical signals around it, which are many, many. All those proteins have to target the correct kind of molecules. They have to be in the right locations. They all have to be present in the first place, or else it just doesn't work. So it's easy to say something like this. Well, some simple cells developed light sensitivity, some spots on them, on the membrane, and that was the start of vision. But you got to get back to that and say, okay, well, what does it take to have a light-sensitive spot? This is what it takes. That's a just so story. Tell me how that happened, and we can go from there. Get down to the biochemistry and say, this is how all that could be in place at the same time for that to work. Otherwise, you're Rudyard Kipling writing a just so story. And that's all it is. And we can make pictures, and we can lay them out in a textbook that is, as far as communication goes, really powerful. But remember this, pictures are not an explanation. They're a story. And if a story doesn't have explanation in it, then it's just a story. And right now, that's all we have from a natural selection perspective on how something like the human eye came to be all on its own and all by itself. The key to this is understanding what does it take to recognize something is designed versus not designed. And you've had that talk. Many of you have heard those kind of talks before. We do this all the time. All the time. Was it murder? Was it an accident? Was it good investing? Or was it insider trading? That's been the news a little bit, hadn't it, lately? A little bit. That was a fortunate decision. 48 hours in advance. Suspicious, right? And we naturally look at things like that and say, that wasn't an accident. That was design. And we do that all the time. We do it in biology all the time. We do it in science all the time. Is that an arrowhead or a rock? All the time. Did you plagiarize that paper? Or did you write it yourself? Imagine, just to illustrate this point, this is a map of rocks in your backyard. Someone took the trouble, for whatever reason, they got a government grant to go map the rocks in your yard. I bet you could do it. I bet you could do it. And they drew a map of rocks in your yard. And this is what they came up with. Just looking at that, there's no reason to think those rocks are where they are because somebody put them there. I don't think. Right? There's no detectable pattern or complexity to it. They're just there. Importantly, the chance of those rocks being as they are is almost infinitely impossible. But yet there they are, but nobody thinks, hey, somebody did that. Pick a rock, I don't care, just look at one, move it the slightest bit to the left. You now have a different arrangement, right? Move it slightly more to the left. And now move it at a 1.2 degree angle to the left. And do that with every rock and every possible combination of moving rocks. You have an infinite number of arrangements rocks could exist in your backyard. Make sense? And yet, the rocks that are in your yard are there as they are, and nobody looks at it and says, you did that on purpose. They're just there. Low probability isn't enough to say somebody did that on purpose. Because almost everything is almost infinitely improbable that it would actually happen. The fact that you are here is incredibly small, and yet here you are. Probability. That the genetics arranged the way they did for you to be here, very, very low probability, and yet there it is. Take these same rocks in the same backyard and rearrange them, however, in another arrangement that is very, very low probability. Something like this. Nobody will look at that and think, well, that's kind of funny how that worked out. They might question the judgment, but no one would question that someone intelligent did that on purpose. maybe as a farce or whatever, but they did it on purpose. And we do this all the time. There is an innate ability to recognize design when we see it. And we do it all the time when we see it. Why not in biology? Why not in biology? Why doesn't that happen? Rocks like this, make it a real case of it. This was no accident, and nobody thinks it was. It's rocks in somebody's former backyard, and people know they put them there on purpose. Right. We infer design because it looks designed, not because we can't figure out any other explanation. Design theorists do not infer design just because natural processes cannot explain the origin of biological systems, but because these systems manifest the distinctive hallmarks of intelligently designed systems. That is, they possess features that in any other realm of experience would trigger the recognition of an intelligent cause. We infer design because it looks designed, not because we're stumped. And we say, well, I don't know how that works. God must have done it. That's not why we make that inference in biology. That's not why we believe. We believe because it looks like somebody did it with immense power and immense intelligence. God is in sight. Why do so many really smart people not see him? Because I know there are some people who may be listening to this who think, yeah, look, okay, I get it, but really smart people disagree with this explanation that I've given tonight. And you're right. Brilliant people, far smarter than me, disagree with this. Why? Why does that happen? This is a quote from a very smart man, belligerent and arrogant, but very smart. Even if there were no actual evidence in favor of the Darwinian theory, we should still be justified in preferring it over all rival theories. Okay, let that sink in a minute. If there were zero evidence for Darwinism, it is still the best explanation that exists on planet Earth. How can you say that? What he's saying is, I don't need any evidence to be a Darwinist. Evidence is irrelevant, is what that statement means. Why? And this is by Richard Dawkins. How can he say that? This is why. This next quote is from a former Harvard evolutionary biologist, Richard Lewontin, hugely famous, huge name in evolutionary biology in the previous century, in the 20th century. And he wrote this in 97, I think, so about 25 years ago. And he's very honest about how this works. We take the side of science in spite of the patent absurdity of some of its constructs, in spite of its failure to fulfill many of its extravagant promises of health and life, in spite of the tolerance of the scientific community for unsubstantiated just-so stories, because we have a prior commitment, a commitment to materialism. We have a commitment, and he's right. Materialism is the belief that nature is all there is. And it's a belief. I mean, no one can prove that, or you can't investigate that. You can't experiment to see if that's true. It's a belief. And Lewontin is right. We have a belief that materialism is true. Well, if materialism is true, then folks, you're never going to see God. Because you've committed, before you look at any evidence, that's an impossible explanation. He is an impossibility. And so Darwinism has to be true. Evidence becomes irrelevant. Lewontin continues. It is not that the methods and the institutions of science somehow compel us to accept a material explanation of the phenomenal world, but on the contrary, that we are forced by our a priori adherence to material causes. A priori means before it starts, at the beginning, we're committed to a material explanation, to create an apparatus of investigation and a set of concepts that produce material explanations no matter how counterintuitive, no matter how mystifying to the uninitiated. We have an a priori commitment to a belief system. Moreover, that materialism is absolute, for we cannot allow a divine foot in the door. that he goes on to say, the scholar who said, if you believe in God, you can believe in anything. They think science is going to stop if you believe in God, which is so demonstrably wrong as to be just absurd. The world is filled today, and history is filled with profoundly successful scientists who had deep faith in God. Why? Because if I take the right point of view, evidence doesn't matter, and I'm never going to see it. It can literally talk to me, and I won't see it. I won't see God, even if he's speaking to me. We see this very literally in Luke chapter 12. Turn with me there, please, in an incident with Jesus, and we're almost done. Luke chapter 12. And Jesus says this in verse 54, Luke 12:54. Then he also said to the multitudes, whenever you see a cloud rising out of the west, immediately you say, a shower is coming, and so it is. And when you see the south wind blow, you say, there will be hot weather, and there is. Hypocrites, you can discern the face of the sky and of the earth, and how is it you do not discern this time? He says, look, you can look at nature, and you can discern certain things. I am God in the flesh, giving literal words to you, and you don't see me. You're feet away from me. Hearing in first person the voice of the Word. Hearing words from the Word. What a remarkable idea that is. And you don't have a clue what you're hearing. I can be like that. I have been like that. Why do people not believe God when they look at biology? Because if you approach it with the right mindset, God could literally stand face to face, toe to toe, and you wouldn't hear him. And that's why. It's the philosophical belief of materialism, which is an absolute commitment. You can't see God. It's impossible. Evidence has become irrelevant. Turn to John chapter 8. You see another similar scenario. John chapter 6, 7, and 8, there's this flow of interaction and conversation that goes on in those chapters. And in chapter 8 and verse 31, Jesus speaking to those who believed in him says these famous words. Then Jesus said to those Jews who believed him, if you abide in my word, you are my disciple indeed, and you shall know the truth, and the truth shall make you free. There's the promise. If you're my disciple, you will hear me, and you will be free. Go down to verse 43 now. Same conversation. This time speaking to those who are his enemies. Why do you not understand my speech? Because you are not able to listen to my word. You are of your father the devil, and the desires of your father you want to do. He was a murderer from the beginning, and does not stand in the truth, because there is no truth in him. When he speaks a lie, he speaks from his own resources, for he is a liar and the father of it. But because I tell the truth, you do not believe in me. So this is another audience, too, as Jesus said. Here's the problem. The problem isn't the message. It's not that the message is veiled or vague or obscure. The message is clear. The problem is you don't want to hear it. And you're never going to hear it. You cannot hear it. Because that's just where you are in your mind. Dangerous place to be. That's where really smart people can be, who do tremendous things scientifically, in the middle of all the speech talking to them and all the language communicating to them, never hear it. It happened when Jesus spoke verbally, and it happens when God speaks through his creation as well. Why believe? Why do we believe? We believe because God is speaking to us. And he's speaking to us in the little bacteria, the little archaea, not bacteria, the little archaea that swim around and react to light and move in response to the light. In the end, whatever anyone thinks about how the world originated, how the origin of species came to be, it all gets back to faith. And we all have a decision to make. We put our faith in materialism and its explanations, which are woefully inaccurate inadequate. Or we put it where the Bible tells us. Hebrews chapter 11 and verse 3. By faith we understand that the worlds were framed by the word of God, so that the things which are seen were not made with things which are visible. It gets back to what I believe. So if you have questions about all of this, well, that's okay. It's completely normal. It's predictable. Voice them. If you're around someone who has questions about these things with faith, listen to them. No judgment, no shock. Let the questions come. God's communication can stand on its own. I've had lots of questions. I still have lots of questions. As a student of biology, I've had sat at the feet of a lot of evolutionists. I've had enough other people be able to help me that I've been able to keep my head where it belongs and keep my faith where it belongs, and it's stronger now than it's ever been. And we need that. And you need to be open with it. And we need to be open to each other with that. Thank you so much for your time tonight.